for the organisers for allowing me to speak today. My name is Kim, and I'd just like to introduce a clinical trial that we're running here at Telethon Kids and to give like a preliminary look at some of the early data from our study. So I'm looking at how water quality might be influencing the development of the infant gut microbiome and how it might be an unrecognised risk factor for gut dysbiosis. So modernisation has brought about numerous benefits. The key one being, of course, is reduced childhood mortality. However, at the same time, we're seeing massive increases in the rates of non-communicable diseases. This graph here is from the Australian Bureau of Statistics, and we can see that over the last couple of decades, there's been massive increases in the rates of food anaphylaxis fatalities. And the red line there is the 0 to 4 years of age. So it's primarily in this early childhood group. These increases in non-communicable diseases have been linked to factors that are known to affect the gut microbiome. For example, we're having increased rates of cesarean sections, increased use of antibiotics, increased sanitation, and overall poorer nutrition and poorer diets. And also, epidemiological studies have shown that there have been increased rates of gut dysbiosis. Now, gut dysbiosis, while it's loosely termed an imbalance or an unhealthy gut, it's defined by two main ways. First being loss of diversity, loss of key species, or a loss of overall function in the gut. And the second way is a colonisation of pathogenic species. Just to give a little bit of a summary of how the infant gut develops, it initially starts right at birth, where the alpha diversity is very low because it's highly specialised to digest milk oligosaccharides. Over the first 3 years of life, the gut continues to grow, continues to get more diverse as the diet gets more complex, until at the end of 3 years of age, when the gut's considered pretty adult-like. So these first 3 years of age are considered the sensitive period of gut development because it's highly plastic and it's easily influenced by a lot of different environmental factors. The key ones being at birth is delivery method, and then it's feeding methods, that's breastfed versus formula-fed, and then the cessation of a milk-based diet into a more complex carbohydrate-heavy diet. In adulthood, the gut should be diverse, rich, and stable, and it only really changes in the case of major diet changes, medical conditions, and long-term antibiotic use. So these first 3 years, it's important that we focus on them to kind of set the scene for a healthy gut into adulthood. While I've mentioned a couple of factors that are key to influencing how the gut develops, there are heaps more. So things like gestational age and early environmental exposures and maternal conditions can all play a role in how the gut develops. However, there are some that haven't been looked at yet. I'd like to raise the question on whether or not water quality might be an unexplored risk factor for gut dysbiosis. So as we know, we add things like chlorine to water to control for microbial contamination and kill off any harmful microorganisms. While this is great and it's been extensively tested to be healthy and safe to drink, it kind of raises the question if this might be acting as a mild antibiotic. Currently, the recommendation is to boil water until around 12 years of age, which would remove chlorine and not exposing infants to chlorine. However, we know now that the gut is still sensitive up until 3 years of age. So this 12-month time point might not be the most appropriate. So we've designed a clinical trial to test some hypotheses. That chlorinated drinking water might alter the development of the infant gut microbiome, and removing chlorine might promote a more diverse gut microbiome, since we're kind of eliminating any mild antibiotic effects through the water. And lastly, a more diverse microbiome might have improved health outcomes, specifically around this allergy and asthma sort of a space. So our clinical trial, we've called it TUMS, is Water Quality and Microbiome Study. It's a 2-group, double-blinded, randomized control trial. Our participants were randomized by delivery method, feeding method, and sex, some of the key confounding factors there, and the sample size was 200. Study duration was 12 months, so we recruited at 6 months of age and kept them in the study for a year. Our study design, participants were recruited during pregnancy at the Joondalup Health Campus in the northern suburbs of Perth here, and we collected a baseline stool sample before fitting participants with either an intervention water filter, which would remove chlorine and remove nitrates and a lot of other things from the water, or they'll be given a placebo water filter, which does not change the chemical composition of the water at all. We encourage participants to use this filter to prepare all of their infants' drinks for 1 year, and then at 18 months of age, we collected one last stool sample. Today, however, since we're still looking at all of the longitudinal data, I'm going to be mainly talking about what we found in the 6-month stool samples and kind of what's going on in this early time point for gut development. So the main features of the 6-month gut microbiome. First of all, infant taxonomy is really highly variable between individuals. If we look at phylum, the majority of infants are abundant in Actinobacteroida. However, there are plenty that don't have it at all or have it in very small amounts. Similarly, when we look at genus, the majority are characterized by Bifidobacterium, but there are some that have none at all. So then I went on to group our participants by feeding method. That's whether or not they're breastfed, formula fed, or a mixed fed, partially breastfed diet, to see what species are emerging there. And we can clearly see that breastfed infants are more abundant in the Bifidobacterium species. Similarly, I looked at this for timing of solid food introduction. Early being before 4 months of age, normal being around 5 to 6 months, and late no solids introduced at all. And the late solid food introductions are higher in those Bifidobacterium species. Next, I looked at the alpha diversity. So this is diversity within a sample. And we can see that feeding method has significant differences in diversity based around their richness and strand diversity for feeding method, where bottle-fed individuals have a higher diversity. And then when we looked at age of solid food introduction, we didn't really see much significance around diversity, except when we compare the Shannon diversity of early and late solid food introduction. We did not see any differences with delivery method in alpha diversity or beta diversity. And again, beta diversity is the difference between samples. There was only a difference for beta diversity for feeding method. So this kind of sums up that the main thing that's affecting what's going on here is the type of the feeding method. Next, I wanted to show you a first look at our longitudinal data. So while we found out that the 6-month gut microbiome is really highly variable, this is a bar graph sorted by the presence of Bifidobacterium. So we can see that Bifidobacterium is really variable in the 6-month group, but in the 18-month, the participants are starting to look much more similar to each other. There are still some that are almost primarily dominated by Bifidobacterium, but we're seeing the rates pretty similar across all the samples. Same with beta diversity. We can really clearly see the difference in beta diversity for the age groups. They become much more similar to each other at 18 months of age, which is in the light blue here, and they're very separate to the 6-month cohort. Same with alpha diversity. It's very clear that the alpha diversity is higher in 18 months of age versus 6 months of age for both richness and diversity. So what's next? Of course, we want to look at this water data. We're still working through the data at the moment, but our research questions would be, does unfiltered water impact gut diversity? Maybe it could reduce diversity by having the chlorine present in the water. And among the unfiltered group, is there a cumulative dose response? Does more chlorine equal more effect? And which water parameters might be influencing certain species? And finally, the important question that we like is, can we see any difference in clinical outcomes between the two groups? Unfortunately, a major caveat of our study is that the allergy questionnaire data is quite minimal, so our sample size is quite small there, but hopefully we can start to see some effect. Our study is using metagenomic sequencing, so hopefully it can allow us to develop a clearer understanding of how the infant gut microbiome develops. And our study will be the first to give a really detailed look at how water might be an unrecognised risk factor for gut dysbiosis. And we'll actually be the first longitudinal intervention study to investigate this. Just like to acknowledge all of the participants in the study and for all of their help. And of course, I'd like to thank my supervisors and my team here at Telethon Kids. Thank you.